The overall goal of this procedure is to assess in vitro the impact of latency reversing agents on HIV mRNA processing. The protocol provides a simple, efficient, and reliable method for assessing simultaneously the effect of LRA on HIV transcription and splicing. This technique provides an insight on the ability of LRAs to induce virus reactivation and clearance of the latent reservoir.
After cultivating cells, place 20,000 of them in 100 microliters of DMEM medium, supplemented with 10% FBS and without antibiotics, in the wells of a 96-well flat-bottom plate for 24 hours. The next day, dilute the dual color reporter construct, TAT and RevDNA, in 50 microliters of serum-free medium, and mix gently. Gently mix the lipid reagent, and then dilute 0.65 microliters of it per reaction in 50 microliters of serum-free medium. Mix gently and incubate at room temperature for 5 minutes. Combine the diluted lipid reagent with the diluted DNA and gently mix. Incubate at room temperature for 20 minutes. Then, dispense 100 microliters of this lipid reagent DNA complex dropwise on top of the cells in each well. Rock the plate gently back and forth to mix, and incubate the plate in a humidified incubator at 37 degrees Celsius with 5% carbon dioxide for 5 hours. After this, transfect additional wells with 100 nanograms of CMV-driven EGFP and DS-Red Express DNA plasmid for compensation purposes.